Okay, so today we're going to talk about, we're going to finish up some of the stuff about import of proteins to the ER, and then we'll just talk about what's actually happening in the ER. Some quick business. There is no recitation next week. Nothing. Don't come Monday, Wednesday, or Friday to recitation. That's because Monday, Tuesday is Rosh Hashanah, so I'm not going to be here, and if I cancel one recitation, I have to cancel them all to keep everyone on schedule. So you guys have all gained some time next week, kind of, because I'm going to actually pre-record at least one lecture, and you guys can watch it online, and that way on Friday, when you come in, we can do some Q&A about it and clarify any questions. What else? For those who have not had recitation yet or who are confused and forgot that you need Scantrons, bring your Scantrons to recitation on Friday or in 2 weeks. And don't forget there is a quiz this weekend. Okay, so far we talked about protein import to the ER. This goes very nicely with the recitation paper for this week. You guys are going to know it backwards and forwards. This is part of the secretory pathway, so I want to clarify over and over again, this is not all proteins that are made. It's only proteins that are in the secretory pathway, which means proteins that are secreted out of the cell, go to the plasma membrane, or go to the lysosome. We talked a little bit about the signal sequence, right, and how you can use microsomes to study the signal sequence hypothesis. And we spoke about some of the basic steps for import, right? SRP, bind signal sequence, brings it to the SRP receptor at the ER, and then it starts to import. And then we started talking about integral membrane proteins, and that's where we left off. Okay, so here's the roadmap again, right? So you have proteins, you know, your mRNA goes into the cytosol, and your proteins need to go somewhere, right? So you can either have proteins that go to the nucleus, mitochondria, wherever, but a lot of proteins go through the secretory pathway, which is ER, Golgi, and then to all these places. And I just have a few figures to kind of show it to you in different ways because I really want you to understand it. So this is a nice one where you're talking, where it starts with rough ER over here. And you see membrane proteins are actually in the ER membrane, and they're transported in vesicles to the Golgi, through the Golgi stacks, to either out of the cell, right, secreted out of the cell, or you could have proteins that are integrated into the plasma membrane because the vesicle will fuse with the membrane, or they can go to the lysosome. So you see why topology, I keep harping on topology, right? So you see why topology is important, right? Especially when we talk about membrane proteins. That's really the only time, right? Who cares about the topology of a protein floating in a vesicle? It's inside of a vesicle. It'll go outside of a cell or into the lysosome. But when we talk about membrane proteins, the inside needs to be on the inside and the outside needs to be on the outside. That's another nice figure showing that, right? So here it's a little bit opposite, but you've got your rough ER up here, and there's your membrane protein, right? So the cytoplasmic side stays cytoplasmic the whole way, right? So your vesicle's budding, here's your cytoplasmic side, and now it's going to fuse with, it fuses with the Golgi, see, cytoplasmic side still outside, and then goes to the plasma membrane, fuses with the plasma membrane, cytoplasmic side, still cytoplasmic side. Maintaining topology the whole way through. Okay, one last way to look at this. And the reason I'm going crazy with this idea is because it's a major concept, and there did seem to be a little bit of confusion the other day when we were talking about the outside and the inside and all that. So this is an experiment you could do to watch this. You can actually label your amino acids radioactively or fluorescently or some way so you can see them, and follow the proteins through the secretory pathway. What do I mean? If you label all your, let's see, you can use S35 label cysteine and methionine, 2 amino acids. And then when translation happens, all of those amino acids, as they're integrated into the protein, are going to be labeled. Okay? And so that's how you can follow them. So this is called a pulse chase experiment. So you pulse them with your label, and then you chase them with something that's not labeled. And it's kind of like you take a little picture of what the cell looks like at that moment in time. So you label for 3 minutes, and all of the cells, let me turn the light off because there's actually some red in this picture. Okay, so you label for 3 minutes, and you can see there's a bunch of things in the ER down here that are, well, it's red in the book, but it's black here. So those are radio-labeled proteins here, okay? And so then you chase, which means you remove this, you wash your cells and remove all the radioactive label and just put normal media back. So no new proteins that are translated will be labeled. Only the proteins that were made in that 3-minute period of time are labeled, and you can follow where they go. So now you can see them, they've gone to the so this is all ER, right? They've gone to the Golgi. Now they're in these secretory vesicles, and now they're being secreted out of the cell. And these are just different chase times. So you would actually have different plates of cells that you would harvest at different times and look at. So you would harvest one at 7 minutes, and so you'd have your time 0 where everything's in the ER, your 7-minute where everything's in the Golgi, and then 120 minutes where you would see things in vesicles or out of the cell. Okay, any questions about this? Okay, did that help people to understand things a little bit more? Some yeses? Okay. Okay, so this is where we left off, and we were talking about start transfer sequences and stop transfer sequences and signal sequences and all these different sequences in proteins that basically determine how they'll be integrated into the ER membrane, right? And so we were talking about, and this is what got us onto the whole conversation of N-terminus or C-terminus being cytosolic. And so you can have it go 2 different ways, right? Either your N-terminus can be cytosolic or your C-terminus. And the orientation of what's in what direction depends on the nearby charged amino acids. So for an N-terminal cytoplasmic tail, you have positively charged amino acids just before the signal sequence. Right? There we go. Positively charged amino acids right over here. And so here's your N-terminal cytoplasmic tail. And then with a C-terminal cytoplasmic tail, you're going to have some positively charged amino acids after your signal sequence. So on either side, it determines which side's going to be out, which side's going to be in. So let's say you wanted to, how could you use this information that I just gave you to create a protein where the N-terminus, let's say you've got this protein right here, right, where the N-terminus is usually on the inside of the ER and the C-terminus on the outside of the ER. Let's say you wanted to change it. What would you do? Let's say you wanted to flip it so the N-terminus was on the outside. What could you do using recombinant DNA technology? So something where you can alter your DNA sequence. What could you do? Anyone want to take a guess? So I just told you that depending on where your charged amino acids are on either side of the signal sequence. So I told you guys, so here's your protein, right? And there's your C, here's your signal sequence. And so I told you if you're positive here, this is going to be on the outside, and if you're positive here, so cytosol. And if it's positive here, then it's going to be, the C-terminus is going to be, that doesn't make sense, C-terminus and cytosolic. So if I wanted you to change something that's C-terminal cytosolic to N-terminal cytosolic, what can you do to your DNA that would do that? So yeah, okay, so I see where you're going. So you said, can you take the reverse complement? Well, no, because actually translation is going to happen in the same way. But I see where you're going because you're talking about flipping it, right? Okay, so where are you guys? Yeah.
Are you just talking about transforming some genetic DNA with a plasmid DNA that codes for an amino acid sequence that's the opposite of what it normally is?
Yeah, change your amino acid sequence. I'm saying, what can you do to your DNA? Because DNA makes RNA, makes protein. Usually when you use recombinant DNA technology in some way to do something like this, you're changing it in the DNA, and you're going to see the change in your protein, right? Because you're not going to put a protein into a cell, you're going to put a plasmid into a cell. So yeah, so you would just change your amino acids so that you can have your positive on this side or on this side, depending on what you want to do. That's how these things are studied, right? How did they find out that depending on where your positive amino acids are, it determines which way things will be oriented? They mutated, deleted, all those kind of things, and saw which way does a protein sit in the membrane, right? Any questions about this? Yes.
So it would be like positive on like both sides of the sequence?
Well, positive on both. I just put it because if it's positive on this side, then the N-terminus is going to be cytoplasmic. If it's positive on this side, the C-terminus is going to be cytoplasmic.
So that means if it's positive on that side, it'll be negative on the other side.
Not necessarily negative, just not positive.
Okay. So neutral.
Maybe neutral. Why not? Yeah. Probably not positive on both sides because that might be complicated and confusing to the cell. Yeah, so the question was, would it be positive on both sides? Usually we'll only have one side being positive, and that's going to determine which way the protein is inserted into the membrane. Okay.
Because like on the slide it says negative, that's why. So I thought.
Right, exactly. So positively charged amino acids following the signal sequence. So positively charged amino acids following the signal sequence, I mean that's going to be a C-terminal. Does that make sense?
Yeah.
Yeah. And when I say following and preceding, the start is N-terminus, right? So preceding would be before the signal sequence, and following would be after, so closer to the C-terminus. Okay, any questions? I will move on. Okay, just a little bit more, another way to look at this. This is just insertion of another protein. It's going to be a receptor. It's a single transmembrane pass protein. And so you have your RNA and your ribosomes, and here's your internal signal sequence. And your internal signal sequence is going to be acting as your start transfer sequence, basically your transmembrane domain. And SRP is going to bind to it, right, because it is a signal sequence. And that's going to bind to your SRP receptor, and then it's going to bind to the translocon, and then you're going to get translation further in, right? So it's coming out of the ribosome further down, so it looks like this. And then then basically you're done, and you have N-terminal on the outside and C-terminal on the inside. The only difference here from between this and, say, a non-transmembrane protein is that the signal sequence isn't cleaved, right? It actually becomes a transmembrane region. Okay, any questions? Okay. Okay, so what would this protein look like? Well, it would be a transmembrane protein. Would its let's see. Would its N-terminus or C-terminus be... Now, I want you to notice there's a signal sequence and a stop transfer sequence. So that's the first thing you should notice. Would its N-terminus or C-terminus be on the outside? Now, you can't tell positive or negative, but it has nothing to do with it because this is not an internal signal sequence, right? So, the way... Were you saying... Someone saying something? The way I would think about this is not easy, right? So the way I would think about it is you have to start to visualize what this would look like coming off of the ribosome, right? First, your signal sequence would come off, and that would be bound by SRP, which would bring it to the SRP receptor, and then it would bind to the inside of the translocon. So it's bound to the inside of the translocon. And then you would get translation of all this sequence into the ER until it hits this, and that would bind to the other side of the translocon. And then it would translate the rest of it, which this would all be outside of the ER, right? And so then it would cleave this off, which would leave it on the inside of the ER. So it would look something like this, right? Where you've got your start transfer sequence and your stop transfer sequence, and that would go and bind to the translocon, right? And then you've got translation of that whole sequence of protein into the ER until you hit your stop transfer sequence. And then your signal peptidase would cleave that, and that would drop down, and that's how it would look. So I just want you guys to kind of practice going through these a little bit in your head. Okay, any questions about how I got to that? Yes.
How did you know it was a transmembrane protein?
How did I know it was a transmembrane protein? Great question. I love these kind of questions. Okay, so remember we talked about hydropathy index and hydropathy plot, and how the more hydrophobic it was, the higher the peak would be. So here's negative and here's positive. The more positive the amino acid, the higher it plots on the graph. Okay? And to be a transmembrane protein, you need to be about 20 to 30 amino acids long. So now we're looking at something that's, well, definitely at least 20 to 30, at least 20 amino acids long, because this is amino acid count. So this is a 650 amino acid protein, right? And then right over here, there's this hydrophobic region. You see that? And it's about 20 to 30 amino acids long, so it's likely to form a transmembrane region. Yes?
Is the N-terminus always synthesized first?
Yes. Yes. So when you have translation, it's always, and that's because of how amino acids look. If you look at an amino acid, you have an N-terminus and a C-terminus, and then as you have each amino acid added on, it's added to the C. It's added to the N. Well, the C is joined to the N. Questions? Is there another one over here somewhere? No? Is everyone happy? Yes?
So you only have one really hydrophobic region. Is that probably always going to be a stop transfer sequence?
So let's say you only have one. So let's say you don't have this at all, okay? And you have this. I would call that like the internal signal sequence / stop transfer sequence. It's only when you have a signal sequence that's N-terminal that you can think of it as a signal sequence that's going to be cleaved. Because if it's in the middle and you cleave it, well, you lose half your protein, which if you think about economy, I always like to think about economy when you're thinking about a cell. Does the cell like to waste energy? Like if you think of like ATP as its currency, does a cell want to waste its currency, its money? No. So it's not going to make a protein and then cleave half of it off and throw it away or whatever happens to it. Okay. Any other questions? Okay. Okay, so let's talk a little bit about membrane protein terminology because it could get confusing. I would put it into two categories. You can have a start transfer signal, which is essentially a signal sequence that also acts as a transmembrane domain if it's internal. And then you can have stop transfer sequences, and that would be a transmembrane domain that might occur after an N-terminal sequence. But I guess don't worry so much about the terminology, worry more about using the information. If I give you a hydropathy plot and ask you something about it, will you be able to use that information? So can one sequence serve multiple functions? Yes, right? Because when you have these internal signal sequence, it's acting as a signal sequence and kind of like a start-stop transfer sequence. And so we just kind of talked about this, right? What determines the topology of the membrane protein? Yes?
Just because you mentioned that you might give us a hydropathy index, can you go back to the one that we were looking at and tell us what it would look like if it was N-terminal cytosolic?
If it was N-terminal cytosolic. Okay. Well, that would be really hard to give to you, actually, because I would have to give you extra information. right? So if it was going to be N-terminal cytosolic, it probably would not have this signal sequence, but just would have an internal. But then I would need to tell you something about the amino acids on either side, because otherwise you're not going to know. Yep, and I probably wouldn't give you names of amino acids because I never told you to memorize all the biochemical properties. I might say positively charged or negatively charged. Okay, good question. Okay, anything else before we keep moving? Okay, so what determines the topology of the membrane protein? I just said it, right? The charge of the amino acids on either side of that internal signal sequence. So why is it important? Why am I harping on topology so much? Why does it matter which way the protein is inserted in the membrane? Like why am I wasting like a lecture and a half or something? Right. If it's not inserted properly, it won't work and then it's useless. Just like the example I gave of, you know, here's your plasma membrane and then there's your antibody. If it's inserted this way, it can nicely bind to antigen and signal an immune response, right? If it's inserted the wrong way, no immune response, right? Okay, so I just want you guys to always big picture. I'm telling you all these little details about inserting proteins into membranes. There's a reason why. Okay. Any other questions about why? If anyone's having a hard time putting this in the big picture, maybe there's something I can explain that would help? No, everyone feels good. Great. Okay. So then there's just double pass membranes where you'll have a stop transfer and a start transfer sequence. And notice they're both internal. So you'll have your start transfer bind and then your stop transfer bind, and that's how it looks, right? So it's a double pass. Notice that you're not cleaving a signal sequence here. And then you can have these multi-pass transmembrane proteins, which is just a combination of starts and stops. Yes?
Why aren't you cleaving the signal sequence of that one?
Because it's not an N-terminal signal sequence. They're two internal sequences.
Oh, okay.
Yep. Yeah.
Both the N-terminus and the C-terminus are?
Yep. Yep. So if there was a hydropathy plot, it would show multiple peaks in the middle. So kind of like this one. This is showing a hydropathy plot of a 7-transmembrane domain protein. Did you guys know there were proteins with 7 transmembrane domains? And now you do. So that's another example. It can get more and more complicated. If I gave this to you on an exam, would I give you the 7 transmembrane domain protein? Probably not, unless I felt angry at you that day. I try to leave my anger outside of a classroom, though. But I am writing the exam in my office. If someone comes in and pisses me off, who knows? Maybe like a 12 transmembrane protein. Okay. So here's another one of multi-transmembrane proteins, just showing how they go in. And it's just these combinations of starts and stops that go into the translocon and are moved over into the membrane, pair by pair. Start, stop, start, stop, start, stop. Move them over, and then you end up in the membrane. Okay, that's it with that. Okay, so what else happens in the ER? So we talked a lot about membrane proteins. There are also GPI anchored proteins. A GPI anchor is a glycosyl phosphatidylinositol anchored protein. So basically the way it works is these anchors are made in the ER, and it's just sitting in the ER membrane. And then you have translation of your protein into the ER, and then there's cleavage at the signal sequence and transfer of the anchor to the protein, or protein to the anchor, I guess you could say. And so you end up with this protein bound to the GPI anchor. So just based on what I just told you, what do you think the destination of a GPI anchored protein would be? Now, if you're wondering why I'm asking destination, then we need to move back for like, I don't know, 2 weeks. Yes?
The ER membrane.
Well, you're in the ER membrane right now. So what's the destination?
Plasma membrane.
Plasma membrane. Great. Great. I knew you wanted to say plasma membrane. Yep. The plasma membrane, because remember, these proteins are going somewhere. There are proteins that live in the ER that make a lot of these things happen in the ER, but most proteins in the ER, it's just a train station to go somewhere else. And I want you guys to remember that because it's a pathway and it's the first stop in the pathway. Okay, so why would you want a GPI anchor? Well, those proteins could easily be released. So you can activate a phospholipase in the plasma membrane, and it will cleave the protein off of the anchor and release it into the extracellular space. Why do you want to do that? Well, it's one way that a signal can be sent out to communicate with other cells. A really cool thing that trypanosomes do. Does anyone know what a trypanosome is? Yeah, it's a parasite. African sleeping sickness. It's a parasite, trypanosomes. So they are really good at evading host defenses, as any good pathogen should be, right? Are you good pathogen if you're not good at evading host defenses? No, you're a horrible pathogen if you're always caught by the immune system, right? You need to be a good pathogen and evade. So trypanosomes actually have a lot of surface antigens that are anchored to their I guess, cell membrane by GPI anchors. And what they do, brilliant, they just release them all. Isn't that amazing? Okay, you want to get me? I'm going to shed all my antigens. Now you can't see me. Now I'm invisible. Very cool. I think it's awesome. Anyways, so that's the blurb about GPI anchors. That happens in the ER. So here's a summary of delivery of proteins to the ER. Right, so you've got targeting, right, SRP, binding signal sequence, or your translated protein bound by chaperones, brought to the ER, association with the translocon, import either post-translationally or co-translationally, and then molecular chaperones on the inside will help it fold. So that's the summary. If you're looking to put it all in some kind of context, go back to the slide and group all the things we talked about into some part of this, okay? Okay, proteins in the ER. What happens? Well, folding, basically, and all these things that need to happen to help it fold. There's disulfide bond formation, which is involved with folding, right? Folding with the aid of chaperones. There's addition and processing of carbohydrates, which continues into the Golgi. Proteolytic cleavage, so sometimes there's maturation of pre-proteins, and that's mostly in the Golgi, though. And then assembly into multi-subunit protein. So when I talk about protein maturation, going from immature, unfolded polypeptide to mature, folded protein that's modified, however it needs to be modified, a lot of that happens in the ER. So it's not just going there for fun. It's not just going there to get somewhere else. It's going there to mature and then get somewhere else. Okay, so here's a nice figure. So you can classify things into chaperones, disulfide bonds, and rotation around peptide bonds, right? So you've got your import into the ER. It associates with chaperones and helps it fold. And then there's something important that needs to happen, right? So it's folding, it's folding, it's folding. If things are good, chaperones release and it goes into a vesicle. This protein that's going into this vesicle, is that a membrane protein? No, thank you. Who thinks it's a membrane protein? Well, of course you're not going to say no. But not a membrane protein. Notice it's not in the membrane. It's just in the lumen of the vesicle. So it's going to be something that's going to be either delivered eventually to the lysosome or secreted out of the cell. So let's say you have a protein and it's folding and it's folding, it's folding, it's bound by chaperones. And is it going to make it out of the ER if it's not properly folded? No. And we're going to go over the mechanism for that. And if it still can't be folded, eventually they give up and it is retrotranslocated, so basically removed from the ER and degraded. Okay? And so we'll go further over that. So what did I say? Chaperones, right? So chaperones not only help this protein get in when it's post-translationally imported, but also there's a whole class, HSP70 family of chaperones. HSP70 means heat shock protein 70. It's a heat shock protein that's 70 kilodaltons. That's its size. So if I say to you, how big is HSP70? I would never do that. It's like, what color was George Washington's white horse? Right? I would never ask you that because that's a dumb question, right? So don't worry about it. But heat shock protein, they're called heat shock proteins because they're stress proteins. If you heat up a cell, you're going to induce the heat shock response, and then it's going to cause transcription of all these genes that are involved in response to stress. And so that's where that name comes from. There's a lot of the chaperones are HSPs, HSP90. How big is HSP90? See how fast you catch on? And so a lot of them are transcribed and expressed in response to stress because what happens when you heat up a cell, proteins tend to misfold, right? Okay, anyways, enough history for you guys. So there's these, so BIP is a HSP70 chaperone that lives in the ER. It's an ER resident protein, right? So that means it always needs to go back to the ER, and we'll talk about how that happens. Sometimes it might go the wrong way, and there's ways to send it back to the ER. And it just, as it's coming off the ribosome and coming into the ER, they bind. There's a bunch of them that will bind to the polypeptide and help it to fold. That's late. Who's trying to get in here? Better be Professor. I just said it better be
Oh, I'm
Professor.
sorry, buddy. Is this Dr. Williams' office?
No.
Oh, I'm
It's a
sorry.
class.
Oh, I'm
Just go over a few.
sorry.
Yes. Looking for Dr. Wimmers. It looked like a tall person, so I opened the door. Not to say a student can't be tall. I'm tall and older than a student. Okay. Anyways. Okay, so I talked about disulfide bonds. So I don't think I said this before, but maybe you knew it or maybe you didn't, but the ER is a highly reducing environment, and that's why disulfide bonds get made there. And PDI, protein disulfide isomerase, catalyzes arrangement of disulfide bonds. So often disulfide bonds will just get made randomly between the cysteines, right? That's a major sulfur containing, right? So it's between two cysteines that are near each other. But PDI will come along and catalyze rearrangement until it's the proper structure. And so that's what disulfide bond formation looks like. Okay, so they're formed and rearranged, and this helps to stabilize the protein structure. And so my question to you is, how come cytosolic proteins don't contain disulfide bonds? I'm telling you now, they do not contain disulfide bonds. Why? I just gave you all the information you need to answer that question, but that also requires you to have absorbed that information. So I said ER is a reducing environment, and that an enzyme that's involved in disulfide bond formation and rearrangement lives in the ER. So why don't cytosolic proteins translated in the cytosol? Remember, what kind of proteins are going to be translated in the cytosol? Proteins going where? Other places, right? Like the nucleus or the mitochondria. So why don't they contain disulfide bonds? Yeah, both of you. Oh no, you guys gonna have to duke it out. Eeny, meeny, miny, mo. Oh.
And it's not reducing environment.
Right, so reducing environment, and there's no PDI. So that's two things. One, chances are no disulfide bonds will be made if it's not a reducing environment. And two, if they are made, which it won't be made, but if they were made, they're not going to be in the right order. They're going to be random and not appropriate because there's no PDI to rearrange them. Okay, so and then I just said, how does the cell make the right disulfide bonds in the in proteins with multiple cysteines, right? I said it kind of is random, right? So a lot of it has to do with having the right free energy and having the right structure. So PDI will keep rearranging until the right structure is obtained. Okay, so then there's this other enzyme that lives in the ER called peptidylprolyl isomerase, and its role is to rotate amino acids around a peptide bond. So it'll rotate around this bond here. So you can see the peptide bond and see how this is, and see it's flipped down here, and that's what it does. Why would it need to do that? Well, because you're folding proteins, often you need to make these kind of adjustments so you get the conformation with the lowest energy. Okay, so peptidylprolyl isomerases also live in the ER. What else happens in the ER? Glycosylation of proteins. So these large oligosaccharides are added to proteins. So they're actually added as a large unit. So there's an enzyme that catalyzes this. It's called oligosaccharyltransferase. And it's actually in the ER membrane. And so you've got this enzyme that makes this sugar, which is just hanging out in the membrane, waiting for an asparagine to add the sugar to. So this becomes an N-linked, because it's asparagine-linked oligosaccharide. A lot of proteins, almost all proteins that go through this pathway end up being glycosylated, also called glycoproteins, proteins with sugars added. So this happens in the ER. Okay? And that's just exactly how the linkage look for those of you who like to look at bonds and whatnot. Okay, here's a practice problem. So you guys can all talk to your partners for a minute, take a little break. Okay, so therapeutic proteins such as antibodies, tissue plasminogen activator, which is an anti-clotting factor, and erythropoietin, which stimulates red blood cell production. They used to be mass-produced in bacteria. A lot of things are mass-produced in bacteria, right? When we talk about drug companies making, like, insulin, let's say. So this process was very inefficient and expensive because the protein would not fold correctly and it would aggregate. This problem was solved by changing the host to cultured mammalian cells. Why did this fix the problem? So based on everything I just told you, you guys have the answer. So talk to your friends. I'll give you guys a few minutes and then we will discuss.
Yeah, thank you. I was like, what's the word?
I just had I did that something just like that. Catalyze bonds.
Somebody was really trying to get him to read this. I hope it wasn't like an emergency. They don't answer every time.
We usually use those because they get called before we run our class ones. They call twice a day.
Okay, since the class is very quiet with this one, I think we'll stop. I think it's partially quiet because half the class isn't here. But I think it's also quiet because maybe people are confused. Who's confused by the question and how to answer it? Or who is like, it's so easy, I don't believe she's making us think about this. Okay. Just making sure. So those of you guys who see people who are not here, you might want to communicate with them that while I really don't care if they're here, they probably won't do well in the class. Because I think not being here, like you lose all opportunities to ask questions and stuff like that. Anyways, okay, so does anyone want to take a stab at how, or like at least start by going through the thinking that process that you went through in getting to your answer?
We said that in bacteria there are no membrane-bound organelles or anything like that, so there is no way to have a specific reducing environment like you would need and the PDI. So that must have been why it was better in the mammalian cells.
Okay, so in bacterial cells, there are no membrane-bound organelles, which means that there's no ER. So there's no reducing environment and all these enzymes that we just went through that are required for proper folding of a protein. So did you want to add something? That was pretty much it, right? So I think in order to tackle this question, you would have to first find out what's the real question, right? Because it seems like it's a little bit hidden in all this information. But really the question is, okay, there are proteins, and now you might even want to think about these proteins. Antibodies, an anti-clotting factor, and erythropoietin. Those are all things that end up being secreted out of a cell. Even if you knew one of those, like antibodies, everyone knows those end up, they're all things that go through the secretory pathway. And then they're not folding properly. So then you should start thinking secretory pathway, not folding properly, bacteria. Why is this not working? And then you start to think, well, what's different about bacteria? And then you get to the no membrane-bound organelles. Yes?
Yeah, to answer the question, instead of talking about the bacteria, actually thought about the mammalian cell, what they had.
Yeah, because it doesn't work in bacteria.
Yeah.
So it's really a compare and contrast. Compare and contrast bacteria and mammalian cells. What's different that would contribute to this working properly, right? Any other questions about how to get through this? I'm really hoping that by the time we have exams, you'll be so used to these kind of questions that you'll be like, oh, that's easy. Okay. Okay, protein folding in the ER. So we're talking about protein folding, and I said something about how if the protein's not folded properly, it will be retained in the ER until it's folded properly, and if it's still not folded properly, it gets kicked out of the ER and degraded. And so one of the ways this happens is through these two proteins, calnexin and calreticulin, and they interact with these sugars. So remember I just said all these proteins are glycosylated. So these blocks of sugars are added to the proteins, and calnexin and calreticulin bind to them, and folding happens, and the sugars are actually altered. And we'll look at that in a minute, and that determines whether or not calnexin and calreticulin can retain them in the ER. But basically, they're retained in the ER until they're folded properly through action with these two proteins. And this is an example in influenza hemagglutinin. It's actually a polypeptide that forms a trimer. So that's three of the identical polypeptides that fold and form a multi-subunit protein complex together, a trimer. And so that has to happen properly for it to make it out of the ER. Okay, so I just said only properly folded and assembled proteins can leave the ER. Okay, so here's an antibody being made, and here it's being bound by a chaperone, and it's helping it to fold and assemble. So this has multiple chains to it, right? There's two heavy chains, and then there are two light chains. So it needs to fold into this multimer, and then once it's folded properly, it can bud out of the cell. Out of the ER. Okay, so proteins that can proteins that can be, whoa, I know it's folded. It's sometimes I'm just like, and I guess forwarded is a real word, so spell check doesn't pick it up. I don't know what forwarded means. Maybe like if you work for the car company Ford, and then they lay you off, you've been forwarded. That was so bad. Thank you for laughing. I don't know. Maybe that'll be an extra credit. Like, come up with a proper meaning of forwarded. Proteins that cannot be folded are removed from by ERAD, which is ER associated degradation. ERAD. I think it's a cool name. And then there's also another. So there's two things that happen. One is a protein that doesn't fold properly. It'll be removed by ERAD. And the other thing that can happen if there's a lot of misfolded proteins and it's overwhelming to the cell, it will activate the unfolded protein response. So that's two things. People often think it's one thing with multiple parts. It's two different things. And that increases transcription of genes and production of proteins that assist in proper folding, such as chaperones, peptidylproteoisomerase, and protein disulfide isomerase. Makes sense, right? Lots of unfolded proteins. Hey, let's make more proteins that will help with folding. Totally logical, right? Okay, so this is the unfolded protein response. So there's this RNA that's just present in the nucleus, and what happens is there's a signal. Let's see. Yeah. So there's a signal that there's an unfolded protein response needs to be activated, and it's unspliced, and it becomes spliced and ligated together, so it's ready. It's like you don't need to do transcription translation. It's just ready. This HAC1 mRNA. It's ready to go when the signal comes in. And you get mRNA comes out, makes HAC1 transcription factor, and that transcription factor then goes back into the nucleus and activates transcription of all of these genes involved in protein folding. That's the unfolded protein response. So just think upregulation of genes involved in folding proteins, right? Okay. Right. So in the ER, you have addition of these sugars, right? We just talked about these N-linked sugars to this asparagine X. X means any amino acid. So X is not part of the letter code. It just means any amino acid. So this consensus, that's where these sugars are added. And those sugars, it's almost time to go. And these sugars are recognized and determine whether or not a protein is going to be kept in the ER or kicked out through ERAD. And we will continue this. Wait, before you go, this is where I'm going to start with the online lecture. So I'm going to go through ERAD, and then I'm going to go through all the snares and RAD proteins and all that stuff to getting to the Golgi. Okay.